Vladimir, I think we can start.
Okay.
The stage is yours.
Okay, thank you. I'm starting.
Thank you.
Okay, I'm trying to change my mode of. Okay. Oh, this one. Hello, everyone. So today I will be talking about intrinsically disordered protein and about how to predict them. And before starting my actual presentation, I will show you most important slides, so which are related to my acknowledgements. So for a long time, I was finishing my presentation by showing a typical slide of listeners of people who helped me working on this particular topic. And eventually I decided that I need to add some faces to this stuff. And I started adding faces, and I collected faces, people who were collaborating with me. And I have quite a few slides which are showing people who are my collaborators. And definitely there are too many of them so to discuss all of their contributions. So, I'm probably will be soon approaching logical picture here. As you can see, it's quite a few. So somebody told me that, so this is my Facebook of collaborators. And I was hoping that I will be able to keep collecting these people. And then in 2016, I published a paper in which I had this almost 2,500 courses. I still had some hopes that it will be possible to do. So then, so in 2019, I published another paper with 1,600 courses. And the final drop was 2021, in which I published a paper with almost 3,000 courses. So definitely now I cannot have pictures of all those people who are working with me. So, but so altogether, I collaborated with more than 12,000 colleagues. So entire, not universe, but so entire planet. So this is my interactome. And stuff which I will be showing you today, it's a reflection of my collaboration. It's not me, it's everybody else. And definitely stuff which I will be representing to you today. So it will not be possible if I will not be able to work with all of those people. So this is my interactome. I'm very thankful to all of these people. So if you do like what I'm talking about today, it's not me, it's all of these people. If you don't like what I'm talking about today, so it's not them, it's me. So please keep the stuff in mind. So and now, so back to my presentation. So I will introduce intrinsic disorder phenomenon. So then I will talk about correlation between intrinsic disorder and protein function. So we'll introduce a structure function continuum. So we'll have a very brief introduction about correlation between intrinsic disorder and human diseases. And then we'll move into how to predict intrinsic disorder. And it's not practical, it's all talking. So, but if you will be interested in doing this stuff, so you can contact me and I can go through all of the stuff with you individually. So introduction of intrinsic disorder. It's pretty straightforward. So everybody knows about protein structure function correlation. It's a classical viewpoint in which protein folds in order to be functional. So in this case, we do have an amino acid sequence which knows some secret pathway how to get folded into a 3D structure. So, and this 3D structure will be responsible for protein function. So as a result, we have this lock and key idea, which was proposed by Emil Fischer in 1893. I'll show the stuff in a second. And then it was a little bit developed further in the middle of last century in order to kind of make correlation between protein structure and protein function, not as rigid as lock and key, so but more flexible. And induced fit was introduced. Now we have this model in which we have glove and hand, which will be kind of analogous to these adjustments. And lock and key once again proposed in 1893 by Fischer. And so as you can see, so there is a kind of a very nice stereochemical matching between substrate and protein active site. So this is how enzyme works. We do have a lot of models which are associated with such kind of representation. And so any biochemistry textbook, while talking about proteins, you'll start with introduction of lock and key. In 57, Koshland made a modification for this induced heat, and he proposed for lock and key, and he assumed that so you have some flexibility at least with an active site, and it's not completely complementary to substrate. So some adjustment would be happening, and as it's shown here, so you do have some changes over there. But still, it's not completely flexible. It's flexible, but not highly dynamic. And entire protein structure still will be crucial for keeping the stuff in place. People were looking at the stuff, and people were applying different theories, and starting from lock and key. And starting from development of this induced feed. People were looking on correlation between catalysis and catalyst, and so it was recognized that so for protein to be active, to act as a catalyst, you need to have a rigid pre-organized active site. And this active site should be complementary to substrate, to transient state of substrate, so to accelerate reaction. And all of the stuff was keeping lock and key at place. Overall idea of protein functionality based on structure function correlation probably can be considered as a big bang of protein science. So because everything which we knew about proteins by the end of last century was based on this idea. As you can see, enzymatic activity, proteomics, protein misfolding disease, 3D structure analysis, everything which is shown on this slide was rooted in this structure function paradigm. However, this classic structure function paradigm works, generally, but somehow not for my protein. And I have pictures here of crying girls and a crying boy, with the question of why. This is actually based on a couple of cases which happened to me in early days. While talking about intrinsic disorder, proteins which are disordered, they are highly flexible. I'll show the stuff in a second. They are flexible. They cannot fold. They cannot get a unique structure. And they cannot be easily crystallized. And some of them cannot be crystallized at all. During my first presentation, in early days presentations, when I mentioned this fact, after lecture, I was approached by a few people in multiple different instances, and they were talking to me about this. They were in structural biology lab, they were asked to crystallize a protein, they failed. And they were told that they're not real scientists because they cannot do a simple task, they cannot crystallize a protein. Their question was, what if it's not me? What if it's my protein? And with very high probability, it wasn't their fault. It was a challenging target. Intrinsically disordered proteins were recognized by examples originally. To me, everything started a long time ago, in around somewhere 1993, 1994. When a colleague of mine came to me to my lab, telling me that I have this funny protein, you can boil it overnight, and actually you can boil it a week. But as soon as you will cool it down, it will be 100% active. He cannot measure concentration of this protein. Looking at him and looking at this protein, I was completely confused. How come you have a protein which can sustain boiling without losing any functionality? And my first question to him was, is it a protein from thermophilic organism? And I was told, no, it's a human protein. Then I assumed that maybe it's not a protein, maybe it's a peptide. I was told it's not a peptide, it's a real protein. And I was shown this gel electrophoresis, as you can see here. It's a not very large protein, but it's a protein, it's not a peptide.
Volodya? We cannot hear you.
So now you can?
Yes, now I can.
Okay. When you lost me?
I lost you when you said when you increase the temperature and then it cools down. It's
Okay. So you can boil it once again, and when you cool it down, it will be still active, and it's not a thermophilic protein; it's a protein from human. Typically, when you are measuring protein concentration, you're using spectrophotometry, so for doing this stuff, so you're looking for absorption around 280 nanometers. And as you can see for this protein, so this flat, it doesn't absorb light, so you cannot measure concentration. So then I assume that so it could be not protein, it's a peptide, and I will show the electrophoresis. Results, and as you can see, it's a protein. It's not very large. It's 14.4 kilodalton, something like that. So but it's a protein. So looking at structure of this protein, so it doesn't have any structure. So it's completely disordered. So then I looked at sequence, and so this is a sequence. So for this protein, so as you can see, it's entirely red, almost. So out of 111 residues, it has multiple glycines, glutamic acid, aspartic acid, and doesn't have any aromatic residues, and have only very low content of hydrophobic residues. So this protein cannot fold. So because it's highly negatively charged at neutral pH, so it doesn't have enough hydrophobicity to attract, to make a hydrophobic collapse. So therefore, it's definitely unfolded because it's kind of poorly charged protein. So so it took me some time to kind of appreciate what I have in hands. So once again, I got this protein around 93. And it took me 6 years. A paper was published in 99. So it took me 6 years to recognize it. So I have something which is interesting and which need to be discussed. So because at that time I was working on protein folding experimentally. So and I knew literature pretty well, and I never saw papers talking about such kind of proteins. It was a very, very unique exception. So so it wasn't just me. People observed these exceptions for pretty long time. So these proteins were discovered and rediscovered many times. So originally, people did not publish papers about these proteins, mostly because of, you have this very well-established paradigm. The structure is responsible for function. Now you have protein which doesn't have structure but has function. So definitely it's not something which kind of a general stream. So, but still somebody published these papers. Each time when they were publishing papers, because nobody was discussing such kind of phenomenon in literature before, so they were given own names and were introducing own terms in order to describe these proteins. So as a result, so by end of last century, we had this intrinsic disorder Babylon, at which we have quite a few papers published talking about intrinsic disorder. So, but nobody recognized that. So this is not an exception, but this is a general phenomenon. So because all of these terms which are shown here, and maybe more than only these terms, so they were used in literature to discuss these proteins. And definitely a person who was talking about vulnerable protein will not be able to understand. So dancing protein or protein clouds or protein chameleon, so will be from similar category. So what happened at the turn of century? So a few groups came with this idea that so what if such kind of proteins are not exceptions? What if we have a new rule? And what if we will have something very interesting? So, and now we recognize that this is definitely the case. So because so after biosynthesis, polypeptide chain can fold, and this protein folding so will be leading to formation of specific structure, and this specific structure will be responsible for specific function. So also proteins can misfold and aggregate, and so for a long time, so this misfolding pathway was pretty well recognized, but was also ignored. So because if you want to deal with protein structure, with folded protein, if you have misfolded protein, so then it will be something out of your interest. So it will be considered as an artifact. So, and approximately 30 years ago, maybe a little bit more, it was recognized that so this protein misfolding is associated with human diseases. And this subject started to be, so protein misfolding started to be extremely popular. So, now we are talking about another pathway, protein non-folding, in which protein is not folded to different degree, and I'll talk about the stuff more later. But still will be functional. So what is also important looking at this picture, so none of these pathways will be at dead end. All of them are talking to each other. Folded proteins can misfold, and this is happening all the time on a regular basis. But we do have a special machinery inside of cell, which will be responsible for keeping proteostasis. And it will be bringing misfolded protein back into functional state, or will be removing this misfolded proteins. Also disordered proteins, some of them should fold or will fold at least partially in order to get function. Upon interaction with binding partners. Some of them can misfold. But also, folded proteins, quite a few folded proteins. So they are not functional until they're at least partially disordered. As a result, we have all of these transitions around the circles in all directions. It makes protein life very interesting and pretty complicated. And it also gives us that we do have quite different types of intrinsic disorder. For foldable protein, it's quite known that so you do have a sequential protein folding, in which you're starting from a folded polypeptide chain. And if you'll start with some folded polypeptide chain, eventually you'll get functional folded protein, so with unique structure. But also you have population of at least transient population of some partially folded intermediates. So this will be in a kinetic pathway. But also you can change environment, you can change conditions, and you can stabilize all of these partially folded intermediates under equilibrium conditions. And for folded proteins, it looks like this. So you do have at least 4 states: a completely unfolded state, so pre-molten globule, a molten globule, and folded state. But once again, all of the stuff will be realized under different conditions. Same chain, polypeptide chain, different environmental conditions, and we have a different degree of folding. So for intrinsically disordered proteins, so it looks like that. So they could be considered in general as polypeptide chain, which is halted at different stages of protein folding. So some of these proteins cannot fold at all, and they will behave as random coil. Some of them will be able to fold up to stage of pre-molten globule. Some of them will be able to fold to molten globules, which means that so they compact and have secondary structure, but still don't have rigid structure. All of the states will be different proteins, but under physiological conditions. So here we have same protein under different conditions. Here we have different proteins under physiological conditions. Once again, we can have native coils, we can have native pre-molten globules, and we can have native molten globules if we are considering entire protein or entire protein domain. Also we recognize that, so we can go and check. How often can you find a protein without any disorder in a protein data bank? And as it's shown here, it's not a very common phenomenon. In protein data bank, which is a place which is collecting information about protein structure. About 30% of proteins in PDB, we'll be looking at this way, so without any intrinsic disorder. But 70% of proteins in general, and actually more than 90% of proteins which are not involved in interaction, which will be structures of just protein folded by itself. All of them will have this different levels of disorder. So in some cases, you can have disordered tails. In some cases, you can have disordered loops. It could be a disordered flexible linker. In some cases, you can have disordered domains. If you're moving away from crystallography, moving into, for example, NMR analysis. In this case, you can see that we can have quite different forms of almost completely disordered proteins. So combining this previous slide in which we are showing that so we have native coils and we have native pre-molten globules and native molten globules. So with this slide, so different parts of protein can be disordered. It can generate us that it can give us an idea that so probably protein structure will be much more sophisticated than it was expected. So I will talk about this stuff once again a little bit later. So, but we have a very high heterogeneity of protein structural organization because of intrinsic disorder. So now you might ask a question: how these proteins look like? So and to look at protein structure, so what typically you will do? You will go to PDB, you will take coordinates, and then you will use programs in order to create images of this protein. And as you can see, depending on what you're looking at, so you can create different views of this protein, so depending on what you're looking for. So so what is important here? So this will be a structure of a protein, so which is actually ensemble average structure, because this is a crystal structure. It's not a single molecule analysis. And crystal is a collection of multiple protein molecules in a crystal. So in one place. So if we have such kind of crisp and very nicely defined structure, so this means that so are all members of ensemble, so they are organized in exactly the same way, and this crystal structure of a single molecule so will be an ensemble average structure of all molecules in the crystal. So this will be structured protein, ordered protein. So if you take into account disordered protein, we'll be ending up with worst case scenario is hairballs. In which protein is not folded at all. And so because of once again, so we are looking on structure which is generated by techniques which are ensemble, which analyze the structure and ensemble. So it's not one molecule, it's multiple molecules are so very overlaid. And so why we have this hairball-like appearance? So this will be because of we do have too many proteins. And each will have individual structure, and the structures will be very different. And if you overlay them, so it will be this kind of pretty undefined and very fuzzy ensemble. So also you can have this situation in which you have, for example, folded protein interacting with disordered protein, and so it's kind of, I'm calling it a bad hair day, and so it's how it can look like. And also it could be in this way. I'll talk about these proteins more so in this P53. So a few years back, I was invited to publish a review about different techniques which can be used for analysis of intrinsically disordered proteins. So I decided I want to illustrate also complexity of these proteins and their structures. And I was trying to publish this pair of structures, P53 and acute kit. Paper was accepted. But then editorial office contacted me and said that so I need to get a permission for reproduction of this cute face. I wasn't able to do it because this is a picture from internet, and I found this llama or camel, and so this structure this pair was published in that review. So, but actually it's a reflection that so you have quite a few disorder. So and it looks like it's bad hair day. So this is another very interesting example. So this will be nuclear pore, and nuclear pore is the only core type of pore which is present in nuclear envelope. It's a very large protein machine, probably one of the largest protein machines, if not the largest protein machine in the cell. And it's organized as this ball of noodles. It has a rigid ball, so which will be making a hole within protein, so within nuclear membrane, and so this hole is filled by, this ball is filled by highly disordered proteins, and altogether, so it will look like this picture which is shown here. And so all of these noodles inside of ball, so will be moving. They will be creating so this highly dynamic mesh, which is preventing molecules from going through this hole. So it will be special carrier protein which will be able to recognize a special protein which need to be delivered inside or outside of nucleus, and it will be able to bring it through this mesh. But so typically, proteins or other molecules cannot go through the stuff spontaneously. Altogether, if you combine the stuff, if you put the stuff together, it will generate this spectroscopy of intrinsic disorder, which will be reflection of structural heterogeneity. And it's shown here in a few examples. So when intrinsic disorder was introduced, we were talking about, so we have structured proteins, which looks like these rocks, and we have disordered proteins, which are completely disordered, which looks like as noodles. And therefore, protein universe can be split in these blue and red categories, or black and white categories. But almost immediately when we start working with intrinsic disorder, so we recognize that it's not a correct representation, because in reality what we have a continuous spectrum of structures, in which we have completely structured proteins on one side and we have completely disordered proteins on another side. But then we have a lot of possibilities in the middle. So because different parts of protein can be disordered, and they can be disordered to different degree. As a result, we have this highly complicated, very sophisticated, highly heterogeneous structural universe for proteins. So not very simple. So now you can ask a question: how these proteins can be manifested? So how disorder can be manifested in proteins? I'm starting with this slide shown here. So these proteins are characterized by turned out response to changes in environment. So what do I mean by that? When you have folded protein, so this folded protein has unique structure under physiological conditions. And if you change the physiological conditions, for example, if you decrease pH, folded protein will be denatured, and in extreme case, it will be unfolded. What does it mean? That if you are decreasing pH, folded protein will be losing structure, so it will be less structured. However, when we have this highly disordered proteins, so they do behave in a very different way. If I can go back to this funny protein, which can be boiled a week and still will be folded, it has very strong negative charge at neutral pH. It has very strong charge repulsion. It cannot fold under these conditions. So it will be highly disordered. It will be behaving as a random coil. But when you're decreasing pH, charge distribution will be changed on the protein, and as a result, proteins start to fold. And instead of getting less structure at extreme conditions at a very low pH, so these proteins start to get more structure, so they start to fold. Which means that the definition of this turnout response to change in pH. Same is also applicable to temperature. Everybody knows that you can unfold or you can denature protein by boiling. When you're increasing temperature, protein will be losing structure. Disordered proteins, they actually start to fold when you're increasing temperature. And this is defined by the fact that these proteins, they depleted in hydrophobic residues, but they're not completely devoiding them. Which means that they do have a low level of hydrophobicity. And hydrophobic interactions are known to increase with temperature. And as a result, when you're increasing temperature, we have stronger hydrophobic attraction, so stronger hydrophobic collapse, and proteins start to fold with higher temperature. Once again, structured proteins will unfold or will denature with temperature, so they will be losing structure. And highly disordered proteins, they start to fold with temperature, so they're gaining more structure. All of the stuff is based on their unique amino acid sequence features and compositions. These disordered proteins are also characterized by multiple other interesting features. For example, binding plasticity. This is illustrated here by protein p53, and I will talk about this protein more later. So it's really very nice, very interesting protein, very interesting example of disordered protein. And what I'm showing here, so P53, it's a very crucial protein which is related to development of cancer. And its natural function is triggering of apoptosis, and apoptosis is controlled cell death in response to damage of DNA. So so this protein is related to cancer development, and as a result, it's considered as a very, very interesting and very important target for analysis in attempt to fight with cancer. So it's a lot of information which you know about P53. So it's probably one of the most studied proteins which we have currently. And what we know about this protein, so it has a DNA binding domain right in the middle here. And it has intrinsically disordered N and C termini. So this protein is involved in a very broad spectrum of interactions, and I will talk about this stuff once again more later. And vast majority of interactions will be happening within this disordered N and C termini of this protein. And if you go to PDB and if you ask a question, what kind of structures are available there for P53? What you will find, so a very large set of structures. None of the structures will be a full-length protein. All of them will be fragments of P53. You can find structures of DNA binding domain interacting with some partners or with DNA, but then we have different short pieces of P53 co-crystallized, so with binding partners. And in this picture, all cyan clouds will be partners of P53, and all differently colored wiglets or worms found within these clouds will be pieces of P53 co-crystallized with binding partners. So what is also very important, so this C-terminal part, which is involved in interaction with multiple binding partners, and as it's shown here, it same region, so which can fold differently upon interaction with different partners. It can gain beta structure, it can fold into alpha helix, or it can stay as a regular structure, so bound to different proteins. So these interactions will be responsible for regulation and control of multiple of these at least four proteins which are shown here, and regulation and control of four pathways. Once again, same region which folds into different structures. And this is completely against of accepted idea that unique sequence is responsible for unique structure, which is responsible for unique function. So now here we have an example in which a unique sequence can fold into different structures, and the different structures will be responsible for different functions. So this is a very important feature of intrinsic disorder. So these proteins also will be can be highly flexible. And structural plasticity will be reflected in this protein chameleon idea. And so this is alpha-synuclein protein, which is related to Parkinson's disease. And I will talk about this protein once again more a little bit later. And as you can see, so this protein actually, so under physiological conditions, it's a random coil, but it can fold into different structures. Some of them will be monomeric, some of them will be oligomeric, some of them will be creating this donut-like entities, like shown here. So they could be aggregates, that could be amorphous aggregates, and also could be amyloid fibrils. And depending on the environment, so this protein can get very, very different structures. And explanation for this behavior is shown on the next slide. So when we have structured protein, so this is a representation of energy distribution for this protein, energy profile. So this is a kind of cut of a very famous energy funnel in which we have a lot of potential structures on top of the stuff, but we have only one structure in the bottom of this energy funnel. So for other proteins, they have unique structure, which is characterized by high conformational stability, by high structural stability, which means also this unique structure is located inside of this very deep and very lowly located energy minimum. These other proteins, so they will show very different energy profile. So for these proteins, as you can see, we don't have so deep energy minimum, so we have cracked, surface. So this surface will be extremely sensitive to environmental conditions, and so proteins can be affected by environment. And as it's shown here, so they can fold. So depending on how the environment will be changing, so these proteins can fold to different degree. Some of them will be able to fall to level of molten globules. Some of them will be able to fall to molten globules. So, but also quite a few highly disordered proteins so were shown to fold completely under specific conditions. So not by themselves, but in presence of specific partners. So and so this will be a behavior of this chameleon behavior of disordered proteins. Then next we need to look at how so intrinsic disorder is encoded in sequence. So, and we'll start with analysis of which was conducted by a colleague of mine, Keith Dunker. What it was done here, so he had a set of proteins which were experimentally shown to be disordered, and we have PDB, so which will be so a place for proteins with structure. And so then he analyzed amino acid composition of disordered proteins in comparison with structured proteins. So using this normalization formula which is shown here, it will be content of given residue in disordered proteins minus content of this residue in globular structural proteins, and this difference is divided by content in globular proteins. So this is normalized relative composition. And because of this difference here, so which means that it might have sign. If my protein, disordered protein, has less particular residues, particular type of residue in comparison with globular proteins, my protein, so this difference will be negative. And if it has more, it will be positive. And if you apply the stuff to set of proteins which were available at that time, so we can see the distribution which is shown here. So we have quite a few residues, which are underrepresented in disordered proteins. They are more present in structured proteins, so therefore they are considered as promoting. So, but also we have a set of residues which are more common in disordered proteins. It will be disorder promoting residues. So other promoting residues will be hydrophobic residues. So disorder promoting residues will be mostly charged and polar residues. So this is kind of grain analysis of amino acid composition. So you can do the stuff in a more combined way. So this will be charge hydropathy plot. So it's actually it was based on this funny protein which was introduced to me in '93, when I was thinking about how common these proteins are, so I decided I need to collect a set of proteins in literature which would be behaving similar to this funny protein, meaning so they are almost completely unfolded, so but do have biological activities. And then I put all of this stuff in this plot. So it's dependence of absolute mean net charge on mean normalized hydrophobicity. So disordered proteins in this plot are located in this very large area above boundary, which separates the structured proteins from disordered proteins, and structured proteins will be located in the corner of this phase diagram. So this picture, so charge hydropathy plot, also can be considered as a disorder predictor tool, and I will talk about this stuff later today. But also, disordered proteins are disordered because they do have high net charge and low hydrophobicity, and folded proteins will be characterized by normal hydrophobicity and not very high net charge. So therefore, to understand if protein is structured to disorder, so a simple analysis can be done. So just calculate mean net charge and compare the stuff with mean normalized hydrophobicity. So and if it will be above this boundary, so protein will be disordered. If it will be below boundary, so it will be structured. All of the stuff gave us a possibility to start developing computational tools, so for prediction of intrinsic disorder. Some first tools were reported early on, but they were not taken seriously because they were based on very specific sets of proteins and were specifically designed for analysis of these sets of proteins. So in 2009, we published a review in which we analyzed, described all of these other predictors which were available at that time, and there were 56. And this number of predictors is continuously increasing. Now we have probably more than 100. Why it's in this way? So it's because of, first of all, intrinsic disorder is highly heterogeneous, so which means that so different groups can use slightly different definitions of intrinsic disorder, and so they will be using these different definitions of intrinsic disorder to create a tool. And also there are quite a few different computational approaches which can be used in order to create a tool. As a result, we do have constant increase in number of these predictors. And definitely another goal will be to try to make disorder predictor more accurate. So, and all of this is responsible for this constant increase in number of predictors. Now when we have disorder predictors in hands, we do have a possibility to ask a question, how common these disorder proteins are. So when I was just starting working on these proteins, so I had one protein in 93. So then, later on, I was given another protein somewhere around 95, 97, so which also was disordered. And then I moved to United States and I started working with alpha-synuclein. So this protein, which is responsible for Parkinson's disease. Once again, I will talk about this protein a little bit later today. So, but it also intrinsically disordered. And so having these three proteins actually triggered my interest to intrinsic disorder. And I started assembling a data set related to intrinsic disorder and coming back to, so it will be this plot. To create this plot, it took me 9 months of very intensive literature search. And eventually I was able to collect 91 disordered protein, which were described in literature as proteins without almost any structure, but with some important biological functions. So, if at that time somebody would ask me a question, how common these proteins are, I would answer that so they are more common than unique exceptions. So because, so definitely it's not one or two, it's more than, so it's about 100. So, but they're also not very common. So because, so I spent 9 months and I was able to find information only about 91 proteins, which means that so they're not extremely abundant. So and I would be completely wrong. So because when we develop computational tools and when we applied them to proteomes, so we found that intrinsic disorder is highly abundant. So what I'm showing in this slide are some results of one of this analysis. So it was repeated. So this was published a long time ago, but it was repeated multiple times using different computational tools. And the picture will be, numbers will be slightly different. So, but idea will be exactly the same as I'm shown here. So what we did, we analyzed archaea, bacteria, single-cell eukaryotes, and multicellular eukaryotes. So this is number of organisms which were analyzed within each category. Those will be a range of proteomes. So this will be average proteome size. And as you can see, so what we have an expected correlation between complexity of organism and size of proteome. And I'll talk about this stuff more later today. So, but less complex organisms, so we'll have smaller proteomes, more complex organisms, I will have larger proteomes. So, but also, so we start to analyze all of these proteomes from viewpoint of intrinsic disorder. So we have possibility to predict percentage of disordered residues for a given protein. And you can extend the stuff to entire proteome. And when you apply the stuff so to entire proteome, what you can see, we have disorder content within proteomes will be ranging from about 10% to about 40–50%. We got extremely excited by this observation, and we're very happy to see that the disorder is not very unique. It's actually pretty common. And so skeptics told us this of kind of having just bulk number of predicted residues. So with an entire proteome, it's not very illustrative. So because what we need to see, we need to look for some functionality potentially associated with this intrinsic disorder, and the functionality should be probably reflected not in the kind of spread distribution of amino acids, disordered residues within sequence, so but they should be somehow correlated with long disordered regions. And as a result, so we kind of reanalyzed all of this stuff, trying to find how many proteins in each and even of the in each of these proteins will have long disordered regions, which are defined as regions longer than 30 residues. And so as you can see, so we have from about, once again, about 10% to about 56 to 70% of proteins. So we'll have long disordered regions. So which means that intrinsic disorder is not just commonly present in proteins, it's also will be assembled in long disordered regions, probably reflecting that it could be somehow related to protein functionality. So so also we have a possibility to ask kind of more global question. I'm not looking for percentage of intrinsic disorder, but looking for if entire protein is expected to be structured or disordered. So it's so-called binary predictors. And once again, I will talk about this stuff later today. And if you'll apply this binary predictors, what you will see, so the completely disordered proteins or natively unfolded proteins. So they're ranging from about 3% to about 42%. And so this bottom line here, so this is a content of intrinsic disorder in human proteome. So as you can see, all of us are highly disordered. We have a lot of disorder. So and if you will take exactly the same data and instead of table put them into a picture, and also we'll add some viruses, so we will end up with this representation. So this is a dependence of average fraction of disordered residues on proteome size. And what we have here, so blue circles, so this will be archaea, green circles will be bacteria, brown squares will be single-cell eukaryotes, and the pink triangles will be multicellular eukaryotes. If you're not considering viruses for now, so just concentrate on this side. What we can see, so to move from less complex organisms, which are archaea and bacteria, into more complex organisms, which are eukaryotes, single-cellular or multicellular. So we definitely do have an increase in proteome size, but also we have a dramatic increase in intrinsic disorder. So which means that so for an organism to be more complex, so it needs to rely on less complex, so intrinsically disordered proteins. We have increasing complexity, so from less complex sequences. And why I'm talking about less complexity of disordered sequences, because so they will be depleted in some residues, will be enriched in other residues, so they will not be using all 20 residues typically, so it will be, so their alphabet will be reduced, and reduction in alphabet will be a reflection of decrease in their complexity. So this side will be related to real organisms, and this left side is a reflection of disorder in viruses. So, and as you can see, viruses have a very wide spread of distribution of intrinsic disorder, and some of viruses will be most structured organisms or entities in this planet, and some of them will be more disordered. So so this is related to so their infectivity and adaptability and everything else. So now, so once again, looking at this picture, so very important claim from here. So all organisms, without any exception, will have a pretty high level of intrinsic disorder. And next question will be why. So and the answer will be probably so for functionality, but because if we have so much of intrinsic disorder, which is present in multiple different proteins, so it could be related to some specific functions which are associated with these proteins. So and so this is definitely the case. So because if you will not consider intrinsic disorder, if you will consider just traditional structure function paradigm, so which is lock and key, so which will be reflected by this upper part of this slide, when we have an amino acid sequence which knows how to fold into specific structure. So this amino acid sequence can solve this folding problem. I will get specific structure, and this specific structure will be responsible for specific functions like catalytic activity or some transport activity. So it will be lock and key and induced feed. So now, if you add possibility for protein to be disordered at different stages of its activity and functionality, as you can see, so you can dramatically increase what proteins can do. So when this slide was created in 2001, people were discussing a few cases of conformational switches in which exactly same amino acid sequence so will fold into slightly different structures in completely different structures and slightly different conditions. So it's, for example, transition from alpha to beta structure. So this will be related to different functionality of these proteins. So now, so this conformational switches extended dramatically. We have information about more than 100 such kind of proteins. They are known as metamorphic proteins, and proteins, same sequence, which will fold in different structure at slightly different conditions. So, but for this stuff to happen, what we need to have this old order, so disorder, new order loop. So disorder is crucial. So for gaining new structure within such kind of metamorphic proteins. Also, we do have another very important pathway, which is shown here. We have proteins, so which are structured, so but which are not functional, and they need to be at least partially disordered or partially unfolded in order to become functional. And so this is really very important pathway, and I will talk about this stuff in a second, because it's a very, very important support of overall idea of intrinsic disorder. And we have multiple functions which will be related to this pathway. So, and all of the stuff will be related to upper half of this picture, which will be related to proteins which potentially can fold into 3D structure. So, even for those proteins, introduction of intrinsic disorder or some level of disorder so will be crucial for their functionality. So in addition to these pathways, we have also a bottom part of this slide, so which will be behavior of proteins which cannot fold. So these amino acid sequences, they are not folding into structures, but they are existing as flexible ensembles. And there are quite a few protein functions which will be heavily dependent on lack of structure. And one of these functions I already showed you, it was nuclear pore. So all of these flexible noodles inside of nuclear pore, so will be members of this entropic activity. So, but also we have another pathway, so in which we have disordered proteins, which will fold upon interaction with specific partners in order to get some function. And because of this pathway, so from disorder to order to become functional, so this pathway was taken by skeptics against intrinsic disorder, claiming that it's not disorder what is functional, it's structure what is functional, and so proteins are not disordered. So this will be proteins which are waiting for partners, and as soon as partner will be present, so they will be functional. So this actually interesting argument, but it's not completely correct. So because these proteins waiting for partner, so they do have own activities. So and their activities will be they're able to recognize a partner, able to interact with this partner, and able to fold specifically into specific structure in order to become functional. So which means that so we do have stuff which is related to disorder, so before appearance of structure. Normal proteins which are described by this upper pathway, so they are able to fold by themselves. They have all information for folding, so encoded in their sequence. So these proteins which are shown here, so which will be disordered but folded upon interaction, or proteins waiting for partners, so they cannot fold by themselves. So their folding code is not complete, and they're completing their folding code by interacting with partners. So which means also at least at the level of sequence, so this will be very different. But also now I'm coming back to this pathway. in which we have completely structured proteins, but which are not functional, which need to be disordered in order to become functional. And we have examples in which protein will be completely disordered, so in order to become functional. In some cases, it will be different parts of proteins, so which will be undergoing this transition from structure to disorder to make protein functional. And so this is actually also very important reflection. So disorder is crucial for functionality. And overall, looking at this picture, so you can clearly appreciate so disorder is crucial for protein functionality. If you will eliminate all red, so from all the stuff, so you can see that so functionality will be pretty limited. So, but also if you will eliminate all blue from the slide, functionality also will be pretty limited. So what does it mean? So for protein to be functional or to protein universe to be uniquely functional, so you need to have combination of intrinsic disorder and structure, and so in different proportions, and all of the stuff will be very crucial for what proteins can do. So when we were looking at what disordered proteins are able to do, so we recognize that so these proteins cannot be enzymes. So because catalytic activity definitely requires stable structure. So you can have some flexibility inside of active site of enzymes. So you can have some flexibility in some loops or in some linkers, so connecting different domains of enzymes. So, but overall structure should be more or less rigid. So if you have intrinsic disorder, so it's not comparable so with catalysis. So which is not completely correct statement. So probably it's corrected kind of stage of current stage of evolution. So but early on very early stages of evolution. So first catalysts, so first proteins, so for catalytic activity. So they do not have unique structure. So and their unique structure is a result of evolution. So but kind of but coming back to what these proteins can do. So they are related to regulation, recognition, and signaling. And so now question is why intrinsic disorder will be crucial for regulation, recognition, and signaling. And I have a few slides to show you so why it's important. So first of all, so for regulation and signaling, so what you need, typically you need to have on-off switch. So which means that so you need to have a complex which will be able to be active or not active. So you need to have highly specific but low affinity interactions. So so high specificity and low affinity. So this cannot be done easily for structured proteins because so high specificity is defined on complementarity in between interfaces, better complementarity, high specificity. So, but also better complementarity will be high affinity. So, which means that so specificity and affinity for structured proteins will be connected. And people are using specificity and affinity as synonyms while talking about structured protein complexes. However, if we have a disordered protein which is able to fold, upon interaction into specific structure, it can form this highly specific complex so with Bayesian partner. So, but because of it was disordered and it folds during protein interactions, so protein-protein interactions, so which means that so some energy was spent for this folding, and as a result, so this this complex will be less stable in comparison with complexes between two structured proteins. So so which means that so definitely so this to have high specificity and low affinity, so we do need to have intrinsic disorder. So another possibility for these proteins will be creating highly dynamic complexes in which no structure is formed whatsoever. So this is illustrated here. So by this case of proteins playing staccato, so which is based on polyelectrostatic model. So in few words, we have structured protein partner which has an active site which is able to interact with phosphorylated serines and threonines in a binding partner. And it's not discriminating between serines and threonines. It can interact with any phosphorylated serine and threonine. And then we have a partner which has multiple phosphorylation sites. So and as it's shown here, each of these phosphorylated sites, each of these phosphorylated serines and threonines, so will have same probability, same affinity for interaction. So which means that at any given moment, so we don't have formation of a stable complex. So we have this staccato type interaction on which protein, zero protein is touching structured protein by multiple binding sites. So complex is formed. So, but no structure is originating from this complex. And so first and second, what is also very important, stability of this complex also will be proportional to degree of phosphorylation. If everything is phosphorylated, so you'll have highest binding. So if you will decrease number of phosphorylated sites by one, so your interactions will be weaker. And in this particular case, so phosphorylated protein has 9 phosphorylated sites. And so from 9 to 5, it will be like real start. So decreasing phosphorylation will be decreasing affinity of complex. And if number of phosphorylated sites will be less than 5, complex will not be formed. No, we have phosphorylation dependent on-off switch. So which means that so for this particular case, so we do have an interesting possibility of not only forming a highly dynamic complex, but also of controlling so strength of this complex by changing phosphorylation degree in one of binding partners. And so once again, this stuff is not possible for structured proteins, so disorder should be present. So if you put all of the stuff together, so we have induced folding, so which will be decoupling of affinity and specificity. So we have proteins playing staccato, so this polyelectrostatic model, which I just discussed, but also we'll have 2 clouds interacting with each other. So it will be an extreme case of this palpation or touching each other type of interaction, and I'll talk about this stuff more because it's directly related to a very important phenomenon which started to be very popular in cellular biology, so liquid-liquid phase separation and membreless organelle formation. But all of the stuff shows why intrinsic disorder is crucial, because it dramatically increases possibility of how proteins can interact with each other and how all of the stuff can be controlled. Another interesting development and important development here will be correlation between intrinsic disorder and alternative splicing. So alternative splicing is a crucial phenomenon which is present in multicellular eukaryotes. So this is actually a way of how and why we have this complex tissue organ organization in multicellular eukaryotes. So each and any cell in our body, so will have exactly the same genome. So, but we do have very different types of organs and tissues. So which means that we do have quite different proteomes. And alternative splicing represents one of the ways of how the stuff can be done. And in few words, what does it mean, alternative splicing? Eukaryotic genes, not all of them, but many, so will have this mosaic structure, in which they will have coding regions, which are shown by these colored blocks, yellow, red, green, and blue. But also they have non-coding regions, so which are shown by black lines. So when this gene is converted into RNA, so typically this RNA will undergo splicing, so which means that all non-coding regions will be removed and all coding regions will be combined together. Now we have mRNA, which contains information for protein to hold, and this for protein. So this mRNA will be used in order to synthesize polypeptide chain, and so we do have all of the segments present here. And so for functional foldable proteins, everything is pretty simple. All the segments within protein sequence so will be responsible for protein folding and somehow related to protein function. So which means that entire sequence is important for the structure. So in addition to normal splicing, we do have alternative splicing, which is illustrated here. As a result of alternative splicing, some coding regions also can be removed together with non-coding regions, so generating alternative mRNA structures, which is shown here, and this alternative mRNA so will create alternative form for protein in which some portion of sequence is not present. Now we have, instead of structure, we do have a very big question mark about how the stuff will fold. So as I mentioned early on, I'm experimentalist, working on who was working on protein folding for pretty long time. So if somebody will ask me a question of what it will be consequences of this alternative splicing for foldable protein, answer will be pretty straightforward. So this protein will not fold. So why? So because sequence is responsible for structure, and if not entire sequence is present, protein will not be able to fold. For foldable proteins. So there are a few exceptions from this rule. If you have multi-domain protein, and if independently folded domain is removed by this alternative splicing, remaining part of proteins will be able to fold and still will be functional. So it will be one case in which alternative splicing will not affect protein folding. If you will remove some loops from protein surface, which are not engaged in formation of specific structure, protein still will be able to fold. Also you can take a few residues from within a globule. Typically not very large number of residues, maybe 5, 8, something like that. 8 maybe will be maximum. Protein still will be able to adjust itself and fold somehow. In all other cases, information for folding is lost, and as a result, protein will not be able to get this unique structure, which is encoded in the entire sequence. If part of sequence is not present, so it doesn't know how to fold correctly. Protein will misfold. And as I will show you later, protein misfolding is directly related to development of various diseases. Which means alternative splicing, which is affecting structured protein and removing pieces of structured protein, will immediately lead to a so-called conformational catastrophe, in which protein cannot fold, will be misfolded, will be related to disease. It doesn't happen. But conundrum here is in a way that we have a very high proportion of proteins which have alternatively spliced isoforms. If you consider entire human proteome, we are talking about 70-75% of proteins in human proteome. We'll have alternatively spliced isoforms. Not all human genes will have this mosaic structure with introns and exons, which are kind of this coding non-coding regions. Some of them will be just one coding region. If you eliminate this simple genes and we'll consider genes which have introns and exons, 90-95% of these genes will be producing alternatively spliced isoforms. So now we have a conundrum. We have a high prevalence of alternative splicing. But alternative splicing can kill protein function, can kill protein folding. Which means that we do have kind of not quite clear situation how the stuff works. And explanation is very simple. Stuff which is alternatively spliced in mRNA, will be mostly coding for intrinsic disorder. As you can see here, completely disordered regions will be about 60%, partially disordered regions will be about 25%, and about 20% will be fully structured. Stuff which is located here will be independently folded domains, flexible loops, and some pieces, short fragments from protein globule. Everything else will be located here. You cannot kill structure if structure is not present. Which means that you can remove any piece of null structure. Remaining null structure still will be null structure. This is very crucial for protein functionality. As it's shown here, using another very interesting protein also related to cancer. This will be breast cancer protein 1, BRCA1. It's a long protein which has almost 2,000 residues. It was heavily analyzed, trying to define structure. And what was found is that it is can-fold. And in the middle, so this middle region, which is 1500 residues, so it's completely disordered. So it contains all post-threshold modification sites, and I will talk about the stuff later in a second. Includes very large number of regions which are responsible for interaction with specific partners. In the bottom of the slide, I have representation of alternatively spliced isoforms. So one is a canonical form, which is entire protein sequence, and all alternatively spliced isoforms also will be having this dotted line, so which will be showing regions which are removed by alternative splicing. So the shaded areas, so will show you how alternative splicing will change functionality of this protein. So and as you can see, by combining alternative splicing with intrinsic disorder, so we can generate a very broad spectrum of protein functions. Without affecting the ability of this protein to fold, we are not affecting foldability, we are affecting functionality. And so this is really very crucial. So and probably it's one of explanations of why we do have more intrinsic disorder, in more complex organisms. So this stuff will be related to this unique use of intrinsic disorder in combination with alternative splice. Another very crucial question will be correlation between intrinsic disorder and post-threshold modifications. So there are more than 300 post-threshold modifications in proteins, and some of them also will be reversible. And one of the most studied and most known post-threshold modifications will be phosphorylation. So in human, we have 520 protein kinases. So this is the largest family of proteins, enzymes with same functions. So they have they're grouped on multiple different groups, on multiple different categories, and so they're working with multiple different partners. And if they're not behaving in the right way, so then we have issues related to multiple human diseases, cancer, neurodegeneration, so behavioral disorders, diabetes, and cardiovascular disease. So really very crucial proteins. Once again, 520. So phosphorylation is important modification, so which is reversible. So which means that we have also proteins which are phosphatases, so which will be removing this modification. So we have about 250 phosphatases. 500 kinases and 250 phosphatases. Why I'm showing the stuff here? So because it will bring us into our next slide. More than 75% of eukaryotic proteins can be phosphorylated. In reality, people who are working with phosphorylation, so they're telling me that it's not quite correct because each and any protein can be phosphorylated. So many proteins can be phosphorylated not at one, but at multiple different sites. So which means we have a very interesting correlation between number of proteins which can phosphorylate or dephosphorylate, number of kinases and phosphatases, and correlation with number of potential targets. If you will use this kind of low ballpark evaluation, 75% of eukaryotic proteins. So in human we have 21,000 proteins. Protein coding genes, 21,000 proteins. If you'll take the stuff, 75% of it, and we'll divide the stuff by a number of kinases, what we will see is that each kinase will serve about 30 substrates. So it's 500 kinases working on the 75% of human genome, human proteome. And each phosphatase will have 61 clients. In reality, it's not correct. Actually, it's very, very low estimate. As I pointed out, each protein can be phosphorylated at multiple different sites. So which means that so it will be not 30, it will be much more. It will not be 60, it will be much more. So if you try to find some structural information about proteins which are phosphorylated and not phosphorylated, so what can you do? you can go to PDB. So once again, it's a data bank which contains information about protein structure. And we can ask a question, how many proteins in PDB so will be present in this phosphorylated and non-phosphorylated form. So same protein. So phosphorylated to non-phosphorylated. And it's not too many. So so now question is why it's the case. And if you don't have too many structures in PDB, there are only two logical explanations for this. So explanation number one, nobody cares. So which means that people are not trying to find structures for this couple of proteins, phosphorylated, non-phosphorylated. Which is not the case because phosphorylation is crucial. So for regulation and so for multiple different activities, it has multiple different consequences, functional and dysfunctional. So which means that people will be trying to find information. So for this phosphorylated, non-phosphorylated protein. Another explanation will be stuff is not easily crystallizable. As I pointed out, if you have disordered protein, so disordered protein cannot be easily crystallized, and probably it will be an explanation why we don't have these pairs present in PDB. So, however, what can you do? You can take structures which are present in PDB, and so you know sites in proteins which are phosphorylated, and you can try to see how the sites are located within protein structures, and what you will find proteins, phosphorylatable sites in proteins will be located in disordered regions, in these regions of missing electron density. So, putting all of the stuff together, it brings us into this very important conclusion. So phosphorylation and likely all other types of enzymatically driven post-translational modifications like acetylation, ubiquitination, proteolytic cleavage, glycosylation. So all of them will be happening mostly in regions of intrinsic disorder. So because these intrinsically disordered regions so will help a target protein to be recognized and to be modified by enzyme. So which means that intrinsic disorder either facilitate or enables binding modification. So it's really very important. Another reflection of importance of intrinsic disorder for functionality so will be given by this protein-protein interaction networks and analysis. And so what I'm showing here, so this will be protein-protein interaction network in yeast. Each point will be a protein, and each link in between proteins will be protein-protein interactions. And as you can see, so this protein-protein interaction network has a highly sophisticated geometry. So it has a very interesting topography. So we have many proteins, so which are involved in interaction with limited number of partners. So, but also we have few proteins which will be involved in multiple interactions. With multiple partners. So these proteins which are highly promiscuous binders which are involved in multiple interactions, so they are known as hub proteins. So now question is, can hubness be encoded by structured proteins? And so do we have a possibility for both hub protein and all of its partners to be from this local key category? And as I'm shown by this slide, so probably it's not a very probable scenario. In reality, when we finished analysis of hub proteins from the point of intrinsic disorder and finished analysis of interactors of hub proteins from the point of intrinsic disorder, so we have found that hub proteins, so which are able to bind to many partners once again, so they will be disordered. If they're not disordered, so their binding partners will be disordered, and it will be a reflection, for example, of phosphorylation kinases and phosphorylatable targets, which I discussed just a few minutes ago. Or we can have intrinsic disorder on both sides. So, and I'm showing here. So we have some hub proteins which are completely disordered, so which are predicted to be completely disordered. So which will be involved in multiple interactions. And we have some other disordered proteins, hub proteins which will have structured domains, but also have disordered domains, and all of the stuff will be related to hubness. and then we might have structured proteins which are not disordered, but which are involved in multiple interactions, and binding partners of these proteins could be disordered. And so this last situation is illustrated by so-called 1433 binding, so 1433 protein, so which is shown here. So it has possibility to interact with phosphorylated binding partners, and these binding partners will be able to adjust themselves within binding sites. So this 1433 protein is highly structured, and in 2004-2005, analysis was conducted for some specific features for binding partners of 1433, and this analysis revealed that partners for 1433 are disordered. So it has about 300 to 400 binding partners, and all of them are disordered. So once again, coming back to this slide here, so disorder is crucial for hubness. If the hub is disordered, or hub will have disordered regions which will be responsible for interaction, or binding partners of hub will be disordered, or both hub and binding partners of hub will be disordered. So disorder is crucial for protein-protein interaction in order to create this very complex protein-protein interaction networks. And it brings us now into this very interesting concept, so which is protein structure function continuum. So so it's based on multiple different pieces of evidence. And evidence number one, proteins are highly heterogeneous from the point of structure. Not only IDP, so but entire protein universe can be considered, any member of protein universe can be considered as this protein with mosaic structure, which has fold-ons, which will be independently foldable units, which are not domains, so because single domain protein might have multiple fold-ons, so this will be smaller pieces. Proteins also might have inducible fold-ons. This will be disordered regions, so which can fold at least partially due to interaction with binding partners or due to changes in environment. Some also could be inducible morphine fold-ons. So it will be disordered regions which can fold differently upon interaction with different partners. And it's kind of this morphine fold-ons were illustrated by behavior of C-terminus of P53. So we also have non-foldeds. These will be regions which are not folded, so but which are functional. We have semi-foldeds, so regions which are always in semi-folded state. And then we'll have unfoldeds, so regions which are structured but not folded, not functional, and need to be unfolded in order to become functional. So what is also important, all of these differently disordered or differently structured pieces can be found within one protein. And what is also important, all of these differently folded or differently disordered pieces might have specific functions. Fold-ons will be related to transport and catalysis. Unfold-ons will be reflection of this dormant disorder. So in order to make protein functional, you need to wake up disorder. So you need to make the stuff unfolded. Inducible fold-ons will be responsible for molecular recognition. Non-fold-ons will be related to entropic chain activities, and semi-fold-ons will be related to signaling and regulation. Once again, all of these different pieces can be found within one protein. So which means that by default, so each and any given protein is not a single trick pony. So it's a highly sophisticated machine, so which because of structural heterogeneity can have multiple different functions. This is actually very interesting consideration here, which tells us a very interesting story. Major part of complexity is coming not from genome variability, but it's coming from variability in proteome. For example, in human genomes, so we have about 21, maybe 25,000 protein encoding genes. But in reality, we have not 1 million, but we have much more than 1 million of different proteins. So differently functional protein, or proteins with different functions. So which means that so from one gene, we can generate more than one sequence. And so this will be a reflection of the statement here that a typical protein so will exist as an ensemble of proteoforms. So which means that so from one gene, so we can have multiple different protein forms, which will be related to different functionalities. So and so this diversity will be related to mutations, will be related to alternative splicing, will be related to post-translational modifications. So as a result, so we do have some basic proteoforms, so which are present everywhere, and so these basic proteoforms are related to protein flexibility. So because even well-folded proteins still will have some flexibility, at least with an active site, and so this actually will be related to what these proteins can do. If you have highly disordered proteins, you have already large conformational ensembles, and so different members of this ensemble might have different functions. So it will be basic proteoforms. So also you have inducible proteoforms, so which will be originating from alterations of RNA, so in the form of alternative splicing or some RNA editing because of mutations, because of post-translational modifications. So and so these inducible proteoforms, so all of them are also dynamic, so which means that so also they are canonical or basic proteoforms. And also another level of complexity will be coming from this functioning proteoforms. So because protein can change structure upon interaction with binding partners. So this can create a new possibility for new functions, which means that some functionality can also change what protein can do. So if you combine all of this stuff together, it brings us into this very important picture here. Instead of one gene coding for one polypeptide chain, so which is responsible for a specific structure needed for some unique function. So when we have a one-to-one correlation, we do have much more sophisticated picture, which is shown on the bottom. So if we will exclude intrinsic disorder from consideration, I will take into account just post-translational modifications and alternative splicing. Even with these modifications, we can have from one gene, we can have multiple different functions related to all of these changes. So, but if you induce intrinsic disorder, in which instead of a unique structure, we have conformational ensembles, so now we can dramatically increase of what protein can do. So definitely, we do have a structure function continuum, so which means that so from one gene, we have an ensemble of structures, and so this ensemble of structures will have an ensemble of functions. And so this is reflected by this firework representation of functionality in the case of p53 protein. So this is one gene. I will have multiple forms related to post-translational modifications, mutations, alternative splicing, and different combinations of these factors. And instead of just one product, so from one gene, so we do have this entire firework view in which we have multiple proteoforms related to functionality of this protein. And it's not only p53, it's applicable to vast majority of other proteins also. So this also brings us into another very interesting consideration, correlation between intrinsic disorder and liquid-liquid phase separation, liquid-liquid phase transitions, and membrane-less organelle formation. So what I'm talking about here. So it's very well recognized now. And so we have a possibility for protein to undergo this liquid-liquid phase separation or liquid-liquid phase transition, in which under some conditions we have normal solution in which so this homogeneous. And then we can change environment, temperature, protein concentration, pH, salinity, osmolarity, binding partner, post-translational modifications, and all of these factors can trigger formation of these droplets. So this droplet will be a result of liquid-liquid phase separation or liquid-liquid phase transition. And so it's not, it's a droplet. It's a liquid inside of liquid. It doesn't have any membrane around. So what it will represent such kind of organization in which So you do have a high concentration of molecules inside and low concentration of these molecules outside. So so this will be related to behavior of many proteins in a cell. So what is recognized now that so cells are inhomogeneously crowded. So what does it mean crowded? It means that so they have a lot a lot of different large molecules. You almost don't have free space. But this crowding is not homogeneous. So you have formation of all of these cellular bodies. So they are known by multiple different names. It will be puncta, it will be foci, it will be bodies, and so many of these bodies will be membranous organelles. So they are formed as a result of highly controlled liquid-liquid phase separation, liquid phase transition. So this will be liquid droplet inside of liquid. So it's water in the water. So it will be different types of water inside and outside. What is also important on why I'm talking about this stuff here, so because all these membranous organelles are enriched in intrinsically disordered proteins, and it seems that intrinsic disorder is crucial for formation, disassembly, and functionality of membranous organelles. So how many of these membranous organelles is shown here? So it's actually a pretty outdated slide. So here we have about 50 membranous organelles. In reality, it's more than 100, probably much more than 100. All of them will be present in eukaryotic cells. Many of them will be present in bacteria. And I'm pretty positive also some of them also can be present in viruses. So they're everywhere. Each time when we have disordered protein, and if you have some possibilities for this protein to undergo this phase transition, we will have membranous organelles. So this slide is a reflection of a crucial role of intrinsic disorder. In 2018, so we conducted this pretty simple analysis. We collected multiple different membranous organelles, so for which composition was known, and we analyzed disorder content for these organelles. And as you can see from this slide, vast majority of them will have proteins with very high disorder content, which is actually somehow a reflection of importance of intrinsic disorder for these organelles. So these membranous organelles are very numerous. So often they will be formed in response to changes in cellular environment. So they are very liquid in nature, which means that so when they are formed, you don't have formation of any stable or don't have formation of many stable interactions. So they will be considered as cross-relates which contain intrinsically disolved proteins and nucleic acids. Lack of stable structure is crucial for fluidity of these membranous organelles. And it also will be very important for them to be functional in this way. They also represent a very interesting case of stability from instability. When we have a complex formed by ordered proteins, we have a brick wall, which is really very stable on one side, but also it's not very stable because if you remove a cornerstone from a brick wall, the entire wall can collapse. In case we have when we have this ball of noodles, which will be disordered proteins, so you can remove as many noodles from this bowl and still will be ball of noodles. It's more stable because of lack of structure, because of lack of stable contacts between members of this ensemble. And everything is related to water. I'm on Florida at this point, and so this is what can be seen in water in Florida. It's not related to membranous organelles, so we have alligators. If you look in some Floridian ponds at night, you will see this nicely looking differently colored dots, which will be reflection of light in eyes of alligators. And why I'm showing this stuff here, because all of the stuff clearly resemble stuff which we can observe also with membranous organelles inside of cell. So therefore, these droplets, which can be identified So, but the entire idea for me to show the slide here, so because so a question is how the stuff is formed and whether it will be a difference in between inside of this droplet and outside of the droplet. So we did some analysis and we have found that water inside of droplets is different from water outside of droplets. And so this could be responsible for how these droplets are formed and so how they are maintained and how they're keeping something inside and how they're keeping so repelling something from the stuff. So we have membrane organelles which are disorder based, so which have also inside changed water, which is crucial functionality of these entities. So now so let's move into some other sites. So what we learned so far. So we have a lot of disordered proteins, and they do have a lot of very important functions. So so question is how they're controlled, and what is recognized now. So these proteins are the very tight control inside of cell. So we have this so-called control chaos. As it was shown. So structured proteins and disordered proteins are different at any given stage of their life in cell. So they do have different coding potential. So typically proteins which are disordered, so they will be coded by genes which have more GNC content in comparison with structured proteins. So we have different transcription rate. So we have different stability of mRNA. We have different translation rate. We have different stability of proteins. So what is interesting about intrinsic disorder? So they will be least biologically stable proteins because they're flexible. They will be easily recognizable by proteases, and they will be digested in a very rapid way. So, but also, so they could form more stable entities. So if they're able to form specific complexes. So one of most long-living proteins in human body, so will be elastin, and elastin has a lifetime of about 70 years, and elastin is forming very sophisticated, highly complex complexes, so highly intertwined complexes. And so this stuff will be responsible for overall stability of this protein. So disordered by itself and highly stable in bound form. And also another interesting disordered protein, which is present in human eye, so will be crystalline. So many crystallines in human eye are disordered. But also so human eye is a unique organ. So because it's not changed. So you know that each and any cell in your body, with the exception of probably brain and eye, will be changed over some period. Eye is different because it has exactly the same content of everything as it was put there during embryo development, which means that life expectancy of eye is life expectancy of human being. These crystallines which are present in eye and which are responsible for keeping our possibility to see, and which are responsible for keeping transparency of matter inside of eye. So many of these crystallines are intrinsically disordered, and they are stable for entire life of centuries. So definitely we do have different levels of stability. In case of elastin, it will be driven by formation of complexes. In case of crystallines, so likely it will be because of special location and special places in which you don't have proteases which will be able to chew them. So if they are controlled, so everything is good. If the disordered proteins are not controlled, we are talking about this square concept, abundance of intrinsically disordered proteins. In human diseases, it will be disorder in disorders. So D square. Disordered proteins are highly regulated and very tightly controlled. So if they're not controlled in the right way, so we have a possibility for them to misbehave. And so regulation can be related to some misbehavior of genetic factors like mutations, chromosomal translocation, alternative splicing, and a level of expression. All of this stuff will be related to some gene alterations. So, but also it could be related to some proteolytic degradation, process-level modifications, or wrong trafficking. So appearance of wrong protein at wrong place at wrong time, it can cause troubles. So, so what I'm talking about, so mutations are simple. It's just something happening to sequence. So chromosomal translocation, which is shown here, so it will be some aberration at pretty high level at which instead of two original genes, so because of translocation, we have two chimeric genes in which so they kind of swapped some of their parts. So this chromosomal translocation is a pretty common phenomenon in cancer, and several very aggressive cancers, especially in childhood, so will be related to this chromosomal translocation. Other alternative splicing, so we already discussed alternative splicing before. So if everything is okay, so we have happy protein. If something would wrong with alternative splicing, and if we're talking about structured proteins, so then we are dealing with so either unhappy or completely not able to functionate protein. So process-level modifications are very crucial. So for regulation of protein activity, so if they're not regulated in the right way, we can have troubles. This is illustrated by protein tau. I will talk about this protein a little bit later. So this is a protein which is microtubule associated protein, which is responsible for controlling microtubules. So and if under normal conditions it's phosphorylated. If it will be overphosphorylated, so then it start to not act as regulator of microtubule. It will fall out of microtubule, start to aggregate by itself, and will be responsible for formation of some inclusions related to Alzheimer's disease. So aberrant proteolytic degradation, so will be another protein also related to Alzheimer's disease. It will be amyloid beta, so which is produced from amyloid beta precursor protein. So this piece is cut by specific proteases, and if it's accumulating at high concentration, once again it start to aggregate, and we have formation of another type of inclusions, which also will be related to Alzheimer's disease. So, and a few words more about Parkinson's disease. So mostly because of it's related to alpha-synuclein. Was actually one of triggering factors for me to move from to move from structured proteins to intrinsically disordered proteins. So Parkinson's disease is the second most common neurodegenerative disease. It's an environmental disorder. And it was shown that propensity for people to develop Parkinson's disease could be related to exposure to some environmental factors, so like pesticides and metals. So this disease is associated with specific death of neurons in specific part of brain, which is known as substantia nigra. So these neurons in this particular part of brain, so they're responsible for producing dopamine, and dopamine is a neurotransmitter, so which is regulating our movements. So death of this dopaminergic neurons also will be decreasing level of dopamine and will be decreasing ability to control and regulate movements. The pathological hallmark of this Parkinson's disease will be accumulation of new bodies and new neurites inside of neurons. So how pathology of Parkinson's disease is manifested is shown here. So you can see that, so this is PET scan, so which is showing activity of different parts of brain, and here, so people are looking for activity of substantia nigra, so this yellow colored blobs are showing that, so in normal brain, so substantia nigra works okay, and in Parkinson's disease, you have dramatic decrease inactivity of substantial nigra. So this is a slice of brain of Parkinsonian patient and person without Parkinson's disease. And so this will be substantial nigra. So this will be region which is producing dopamine. And as you can see, it's almost absent in Parkinson's disease. And if you look under microscope, so for these samples, what you will see that in Parkinson's disease, you almost don't have these brown neurons, which are dopaminergic neurons. So it's almost completely gone. So neurons which survive in Parkinson's disease, so they will have the specific inclusions, so which are Lewy bodies. So this will be a very interesting correlation between this is so they contain aggregated protein. So we also have during neurites, it will be also aggregated protein which is located inside of axons, and axons looks like clogged by this aggregated material. Major player in information of which Lewy bodies will be protein known as alpha-synuclein. So correlation between alpha-synuclein and Parkinson's disease was found in 1997 when it was shown for 3 families where there are specific mutations in gene encoding for alpha-synuclein. And so these people with these mutations in gene were kind of the families with early onset familial form of Parkinson's disease. So when people start to analyze inclusions in Lewy bodies and Lewy neurites in Parkinson's disease, and they have found that alpha-synuclein is commonly present here. It was 97, and now we recognize that so it's not only Parkinson's disease. So we have this aggregated alpha-synuclein present in multiple different diseases. We are talking about synucleinopathies. And I have next slide here, which is showing how many of these synucleinopathies are present. And as you can see, it's quite a few diseases, and they are affecting very different regions of brain. So, but in all cases, what we have accumulation of misbehaving, misfolded, aggregated alpha-synuclein, which will be causing to all of these neurodegenerative diseases. What is so special about alpha-synuclein? So this is a protein which has multiple material spliced isoforms. It has multiple phosphorylated modifications. It can interact with multiple partners. It's predicted to be disordered, and it's shown to be disordered experimentally. It has multiple disorder-based interaction sites. It has these molecular recognition features. So these are disorder-based segments which can fold upon interaction with binding partners. It has multiple phosphorylated modifications, and is predicted as disordered or was mostly disordered by many different tools. It has strong propensity for liquid-liquid phase separation, which can be evaluated computationally using this postdoc platform. As you can see, it has a droplet promoting region, and it has a high potential for phase separation, which is high value for probability for liquid-liquid phase separation. It also acts as a half protein. Alpha-synuclein here interacts with all of these proteins around, it's a protein chameleon. And actually, entire idea of protein chameleon was derived from analysis of alpha-synuclein, so which is able to fold into different structures depending on the environment and depending on binding partners. But what is also very interesting, it's still considered as a protein with unknown function. And I spent a little bit more time here. So how come? As you can see, there are probably close to 25,000 publications now talking about alpha-synuclein. So this stuff was published in 2017, and at that time we had 16,000 papers. And the vast majority of these papers would start with claim that alpha-synuclein is a presynaptic intrinsically disordered protein, so with unknown function. Many not vast majority, but at least pretty large portion of this papers. So we'll be discussing some functionality related to alpha-synuclein. So, which means that so for one protein we have multiple different functions, but you don't have one function. Because of all of us are continue to be trained, so using this lock and key idea in which we have one sequence, one structure, one function correlation. If you don't have one unique function, so it will be considered as function is unknown. So in reality, this protein has multiple functions. It's also related to multiple pathologies. And it's a highly abundant protein. So estimates are that about 1% of all soluble proteins in brain, so will be alpha-synuclein. It's everywhere in brain. It has multiple different functions. And if it's not behaving in the right way, so we have multiple possibilities for development of multiple different diseases. So this is kind of funny representation of functionality of intrinsic disorder. So this is an actual structure of alpha-synuclein bound to vesicle, and as you can see, it has a structure so which resembles this octopus. It's one protein, highly flexible. As a result, looks like it has multiple arms, and because of the stuff, it has multiple possibilities for different activities. When this protein start to form aggregates or oligomer, so we have even more possibilities for creating some activities or unwanted activities for this protein. Multipathogeneity, multifunctionality, connection to different other diseases. So for alpha-synuclein, so will be directly related to this proteoform concept, in which from one gene, because of different proteoforms, we have multiple different protein forms, so which will be related to whatever this protein can do in the right way, and also to whatever this protein can do in the wrong way. So definitely proteoforms are crucial for explanation of all this interesting behavior of this interesting protein. My next candidate will be P53. So I already showed you this picture before. So this will be the structure of P53. So real structure is shown by this surface representation, and all of these wiggly tails, it's modeled pieces added. And so this is just N-terminal part, and similar fuzzy set of tails, so will be on the bottom of this picture, which will be C-terminal part of this protein. So we can go to AlphaFold and look at structure which is generated by AlphaFold to P53. And as you can see, so it has structured domain, but it has also a lot of disorder. So how to read AlphaFold related structure? If you have blue, it's a very high confidence of model. If you have cyan, it's confident. If it's yellow and orange, it's low or very low confidence. And when you have low and very low confidence, so claim is that so these regions could be disordered. So also keep in mind so that this is just a snapshot. So this is not an entire P50. So this is not an entire ensemble of structures for P53, just one member of this ensemble. We can take this nice tool, which is D2P2, so for analysis of intrinsic disorder in proteins, and it's predicted to be pretty disordered by multiple different tools. So it shows pretty good agreement between multiple different tools. It has multiple disorder-based binding sites, and I'll talk about the stuff a little bit later. And already talked about the stuff before, and it also has multiple post-translational modifications. So this protein actually shows us a very nice illustration of functional and structural complexity of intrinsic disorder. So you already saw this slide before. Also related to functionality and related to structural plasticity. And so once again, I showed this slide before. We have a region which is responsible for interaction with multiple different partners and falls differently upon interaction with multiple different partners. It's a protein hub. It's right in the middle. So this one over there that has multiple binding partners. So what is also important, it belongs to category of don't touch me. As I pointed out before, so P53 is a protein which is related to multiple cancers. So people are saying that so more than 50% of human cancers will be associated with misbehavior of P53. Or associated with mutations of P53. This top plot shows disorder distribution for P53 evaluated by multiple different predictors. So they're showing pretty good agreements. We have structured domain, we have disordered regions. So this middle part shows position of post-translational modifications. Gray bars show positions of disorder-based binding sites, these molecular recognition features. And so this bottom picture here shows a distribution of mutations in P53, so which are associated with different cancers. And so this protein has 393 residues, and only 3 of these residues were not associated with cancer. So 390 residues of P53, if they are mutated, so they will be related to one or another form of cancer. So this is definitely from the category don't touch me. So it's so crucial, so important. Any piece of this protein is important for normal functionality. If something is changed, it will not be behaving in the right. So it has multiple alternative spliced isoforms, and these alternative spliced isoforms will be affecting functionality of this protein. So you have another partner related to P53, so which will be MDM2, and MDM2 so it's a protein base which is responsible for ubiquitination of P53, and when P53 is ubiquitinated, so it's digested by proteasome, and it's not acting as a regulator of apoptosis, and as a result, so cancerous cells are immortal. So this MDM2, it has a lot of disorder by itself. But it's involved in interaction with P53 and regulates once again. And so MDM2 is also a protein hub. Now we are moving into last part of my presentation today, about how to predict intrinsic disorder. We want to discriminate in between these hairballs and protein with crisp structure. If amino acid sequence is responsible for protein structure, it's also responsible for lack of protein structure. So which means that we do have structure and disorder are both encoded in amino acid sequence. But language of coding for structure and disorder should be different. If this is the case, so then we will be able to create computational tools which will be able to discriminate in between structure and disorder. How we can be sure that structure and disorder proteins are different? So from viewpoint of sequence, I already showed you the slides before. I'll repeat them once again. At global level, highly disordered proteins will be characterized by this combination of high absolute mean net charge and low hydrophobicity. So they will be located in this big area of place diagram connecting charge and hydrophobicity. Structural proteins will be located in the corner over there, and we can have a boundary which is separating the structural proteins and disordered proteins. So this is at very global level. Highly disordered proteins are depleted in hydrophobic residues and highly enriched in charged and polar residues. So this is further illustrated by this picture which I already showed you before, this composition profile, which shows that we have residues which are mostly hydrophobic, which will be depleted in disordered proteins which are related to order. And we have residues which are polar and charged, so which will be related to lack of structure to disorder, so disorder promoting. So once again, if sequences are so different, so computational tools can be created to use these differences in order to analyze or to predict intrinsic disorder. So before doing the stuff, we need to talk a little bit more about why we need to have multiple different tools for analysis of intrinsic disorder. And the claim is here. We have continuous spectrum of and in-depth of intrinsic disorder. What it will be the best way to analyze such kind of proteins. If we are using just one tool, we kind of in the case of this 5 blind men and elephant situation. Probably you're familiar with this concept. 5 blind men were asked to describe an elephant, and each of them were touching a particular body part of elephant. And then they start to fight about how elephant look like, because all of them had some experience with touching a particular part of elephant, but they did not see entire picture. Overall idea from this slide, in order to see an elephant, you need to have a possibility to touch different body parts in order to be able to create a more general picture. How you can look at IDPs? You can use one eye, using one technique. You can use one eye with some magnifying glass to see better. You can use two eyes with binocular. You can have 8 eyes, as it's shown here for spider, or you can have a complex eye, which is shown for a fly. And this complex eye is a very interesting idea, because it actually provides a possibility to have a very large view angle and ability to detect fast movements. So this is exactly what we need when we are looking for intrinsic disorder. So what does it mean? So we need to have a combination of multiple different tools, which will be able to be combined with this complex eye to see intrinsic disorder better. Once again, instead of looking, we can have this multi-tool, which is specific for multiple different levels of intrinsic disorder. But all of these tools will not be completely accurate because they will be missing something. And typically, you need to have a combination of multiple tools in order to describe intrinsic disorder in the best way. This is applicable to both experimental and also to computational description. Probably I can skip it and go directly here. How you can create a tool for predicting intrinsic disorder? You need to start with a disordered sequence data set. You need to split this data set. You need to extract specific attributes related to intrinsic disorder. It could be related to composition, it could be related to content, it could be related to multiple different factors. Then you need to split your set of disordered proteins and set of structured proteins into training and testing set, and then you can predict your train your predictor. Then you do validation, and do out of sample data analysis, and then you will have your predictor. This is actually a very, very crude representation of how the stuff works. Overall, we'll be coming back to this idea, that amino acid sequence codes for both structure and disorder. If it's the case, then we can develop computational tools for looking for disorder. And typically, what you will have, for such kind of predictors, you'll have a sequence, and the sequence will be looking for composition, sequence complexity, heterogeneity, charge, and multiple different other factors. And you can have all of these factors analyzed within a sequence using sliding window, and this window can range from 15 to 60 residues. When first predictors were developed, there was this predictors of naturally disordered regions or ponders. They were showing some accuracy, around 70%. Now it's approaching 80-85%, and probably some of new generation predictors, will be close to 90% from the point of accuracy. And I showed this slide before. We started with 1 or 2 predictors. Now we have many more, and all of these different predictors. They are available or many of them, so information about them are available on this DISPROT database. It's a special database for intrinsically disordered proteins. So and so once again, it's quite a few disorder predictors around. So now let's talk about different types of disorder predictors. So we are starting with a very simple one, so which is scale. So what does it mean, scale? So it's very simple. So scale means so for each residue, so we are given a particular number, and so we're looking at amino acid sequence from this a particular fixed number which is assigned to a particular residue. So and there are multiple ways how this value can be assigned. So one of first such kind of tools was developed by a group of Oksana Galzitskaya. So what they did, so they looked for a number of physical contacts within protein structure for each residue, and they calculated number of contacts, and they kind of for each residue, so they produce this an average number of contacts. And now, so you can take these numbers, and so for foldable proteins, you have a particular expected number of contacts, which will be reflection of presence of a stable structure. And if you have number of contacts which will be below this value, so it will be kind of a reflection of the support and probably will not be folded. So this will be related to number of contacts. So and it's resulted in the development of this fold-unfold predictor, so which was trained on 427 native unfolded proteins and about 560 globular proteins, and it shows that so it's pretty accurate. So they're using different windows. So another approach, stop IDP scale. So here, so we looked at 517 different amino acid scales, and we looked for different combinations in between amino acid scales in order to find the best combination which will provide best separation in between structured and disordered proteins. And so the stuff was found, and so now it's called top IDP. It gives us best separation. So and each amino acid, so within the scale, so will be assigned a particular number, and so this normalized unfoldability. So tryptophan so will be located on left side, so it shows that so it's not very probable to be disordered, and proline will be located on the right side, which is a reflection of so it's most probable residue for being in disordered region or in disordered protein, and all other residues will be located in between. So this will be scale. Each residue, so you'll have a particular number. So what can you do? You can take this so scale-based approach and can move them into super residue predictor, which is shown here. So for each residue within sequence, we are given a number which is shown over there, or which is shown over there, and so then we can look at the stuff using sliding window, and so we can generate a disorder profile, and once again it will be bringing us into category of residue disorder predictors. So so this is illustration of application of one of these disorder predictors, a series of these predictors for one of proteins. So and how you can prove validity of your prediction. So the stuff can be done experimentally, and how it was done here for this XPA protein. So protein is predicted to have disordered N terminus and disordered C terminus, and also structured middle domain. Also, so you can predict accessibility of different. First of all, you can predict potential cleavage site for different proteases. And the idea of proteolysis is very simple. So protease will digest regions which are accessible. So this bars in the middle, so they're showing all potential cleavage sites. So for this XPA protein, so by different proteases. So, and then protein was subjected to proteolytic cleavage, and we were able to find which residues were used proteases by cleavage and which were not accessible. And so stuff which were digested are shown by red bars, and stuff which wasn't digested so shown by blue bars. And as you can see, we have a very good correlation between disorder prediction and accessibility to proteases. So once again, stuff which is disordered is accessible, and stuff which is structured is not accessible to proteases. So we do have multiple members of this order family. So so it's using window, so ranging from 9 to 21 amino acids. So they're using multiple attributes. Fractional composition of particular amino acid residue, hydropathy, sequence complexity, and so they're using in multiple different combinations. So the stuff is used for making prediction. Now we are moving into another level of another way of predictors, which are binary disorder predictors. So what does it mean? So here, we are making prediction for entire protein, and it will generate a protein profile in which you can see how disorder is distributed within sequence. So you can see structured regions, and you can see disordered protein, disordered regions. Binary predictors are operating in a different way. So binary predictor is just giving you a possibility to give an answer yes or no for protein and for question structured or disordered. So binary predictor will tell you if your protein is expected to be structured or is expected to be disordered. So one of binary predictors will be this charge hydropathy plot. So I showed this stuff already before. So what you're looking at absolute normalized net charge dependence on mean normalized hydrophobicity. If your protein is located above this boundary in this upper level of this phase diagram, so it will be expected to be disordered. If it's located below this boundary, it will be expected to be structured. And so this boundary is used as this binary disorder predictor. Another way will be to look for this pair residue disorder predictors to convert them into binary predictors. And this stuff is done by creating this cumulative distribution function analysis. How it works is shown here. So what we are looking for cumulative fraction of residues, so with particular disorder score. If protein is completely structured, all of disorder scores will be 0, and as a result, this line will be kind of coming back on this upper square, upper corner. If protein is completely disordered, all of its residues will be predicted to have a disorder score of 1, and it will be creating this line coming in this back corner. In reality, proteins are mixed. We have structured and we have disordered. And so we have experimentally validated proteins, which are structured, shown by blue lines, and which are disordered, shown by red lines. We do have, you can see that they're definitely different in this representation. And we have a boundary line. And when all of the stuff was analyzed, it was recognized that it's enough to have this boundary line, with 7 points, in order to discriminate between structured and disordered proteins. And discrimination is based on majority voting. If 4 points of your CDF plot are allocated above boundary, so protein will be disordered, at least 4. If at least 4 points in your CDF plot, CDF curve will be located below boundary, so protein will be disordered. What is also interesting, you can take binary predictor and convert the stuff into pair residue predictor, and it was done for CH plot analysis. Instead of taking entire protein, exactly same procedure. Calculating mean disorder, calculating absolute mean charge and anhydropathy is applied to different parts of protein, and sliding window is used, and you can generate this fold index profile, which is based on charge hydropathy plot. As I pointed out, so it's best way to look at proteins. So will be to use not one but multiple different disorder predictors. So because different predictors will be sensitive to different parts of proteins, and this is very nicely illustrated by this combination of charge hydropathy with CDF. Bond-based CDF so will be able to discriminate in between structured proteins and all types of intrinsic disorder. So therefore, we have this brick wall placed in between structure and disorder. So on the other hand, charge hydropathy plot so we'll be able to discriminate in between highly extended, highly disordered proteins, structured proteins, and native molten globules. Native molten globules are compact, so which means that they do have a high enough hydrophobicity level. So they will be combined together with structured proteins. So and the brick wall is here in between highly disordered proteins and molten globules and more structured proteins. And if you combine the stuff together, as you can see, we can produce much better representation of intrinsic disorder and better understand intrinsic disorder. So how it works? For CDF analysis, as I pointed out, so we have this boundary which is separating structure and disorder. And so for query protein, so we are calculating a mean distance of CDF curve for this boundary. So it will be an average distance between the 7 points. So for structured proteins, so difference will be positive. So for disordered proteins, difference will be negative because so CDF curve for disordered proteins is located below boundary. So for charge hydropathy analysis, it's even simpler because each protein is present in the form of 1 point in this charge hydropathy plot. And what we are doing, we are just measuring the distance in between plot and so charge this boundary line. So we have an average value here, so we have value here. So you put it into this delta CDF dependence on delta charge hydropathy dependence on delta CDF, and we can get this picture which is shown here. So we have structured proteins will be located in bottom right corner. So proteins which are compact, so but disordered or contains a mixed content, high content of both structure and disorder will be located in bottom left corner. Regions which are located, so proteins which are completely disordered, predicted by both predictors, will be located in upper right corner. And here will be proteins which are predicted to be disordered by charge hydropathy, but structured by CDF. So which are kind of we are kind of calling them disordered strange proteins because there are not too many of those, and they're not quite clear who they are. So if you will take this and if you will apply it to entire proteome, what we will see, we will have this picture here. So this is this CHCDF analysis. So for mouse proteome, and as you can see in mouse, we have 55% of proteins are predicted to be completely disordered. We have about 30% of proteins, so which will be either hybrid proteins or native molten globules. We have 12% of proteins, so which are expected to be completely disordered. So and once again, these are binary predictors. We are not talking about percentage of disorder. We are talking about overall expectation of disorder for these proteins. And we have about 2.5% in this upper right corner, so which will be strange proteins. So what we know about proteins here, so they typically will be quite often will be highly enriched in cysteines, and cysteines are treated in different ways by different predictors. Some of predictors will define cysteines as most odor promoting residues. Other predictors will look at their, for example, hydrophobicity and based on hydrophobicity, so since are not most hydrophobic residues, so will not be how most disorder promotes residues. So anyway, so it's a very useful tool which will allow you to have a kind of interesting overview of overall disorder status of entire proteome. So now, so we do have application of the stuff for multiple data sets. And so what it was done here, so we analyzed HIV proteins. So HIV, so there are multiple strains of HIV, so there are multiple forms of HIV proteins associated with the different strains. And so proteins will have some specific activities. And from the point of this CHCDF analysis, as you can see, what we have some proteins which are specific viral enzymes crucial for viral activities. They will be located in this quadrant responsible for structured proteins. So, but then also you'll have a lot of proteins which are completely disordered, and these proteins will be related to communication of virus with host. These proteins will be responsible for convincing host to act as a factory production of everything which is required for virus. So so this is actually will be done by mostly disordered proteins. So and it brings us into last part of so predictors, which will be meta predictors, and by default meta predictor. So it's a set of different predictors combined together. So they will be so outputs of different predictors will be used as input for this another predictor. And so best way of looking at this stuff, it will be once again coming back to elephant story. So best tools so which could be combined for this meta predictor. So will be predictors which are looking for very different categories or very different parts of elephant. So predictors which are included in this meta predictor should be complementary to each other, not orthogonal, not parallel. So and the accuracy of this meta predictor will increase dramatically if you have this orthogonal way of looking at complex. One of interesting examples of this meta predictor will be ponder fit, which was assembled from multiple different ponders. Also IUPRET, fold index, and top IDP were included. As it's shown here, so this ponder fit showed overall increase in accuracy from 3 to 20% in comparison with individual predictors. So what is also important, and why I'm showing this stuff here, so one of tools which was used for training of ponder fit, it was the top IDP. So and top IDP is the least accurate predictor. Actually, it's not very accurate at all. When we were trying to see how important top IDP is for accuracy of ponder fit, so we kind of how the stuff was done, we were taken out of these different predictors one by one and seeing how the stuff will affect accuracy of disorder predictor, results in disorder predictor. What we have found, so when top IDP was taken out, accuracy of ponder fit dropped dramatically. So this is a very important observation here. So we do have a tool, so which is not accurate, so but which has a very unique view at intrinsic disorder, a very unique view at problem, so which we're analyzing. And even if it's not very accurate but very unique, it still adds a very important piece of information in order to make prediction better. So we also have this readout tool, which is publicly available at this URL over there. And so it combines 6 established disorder predictors and in unique single unified platform. It generates parallel predictors. It generates CHCDF analysis. So for a protein, it's applicable for single protein. It's applicable for data sets. It can handle probably up to 30,000 sequences at once, and it's pretty fast and actually very useful. And once again, it gives you an output of multiple predictors in one place, so which will give you a better understanding of how disorder is distributed within your protein. So we have some other disorder predictors, meta predictors. Once again, meta predictors, they evolved various sources of information, predominantly obtained from orthogonal prediction methods. And we have all of these different disorder predictors, which will be from this category. And I was fast because I'm almost done. We have intrinsically disordered proteins, which is a new concept in protein science. And so it's very crucial because it gives you a possibility to answer to multiple questions which were not answerable using structure. It opens a new way to look at how protein looks like and what protein can do. And it's definitely a very interesting development. But it's not in a way that disorder is better than structure. It's in a way that we need to have both, because disorder and structures will be complementary. Disordered proteins can do stuff which cannot be done by structured proteins. Structured proteins can do what cannot be done by disordered proteins. And only in the case when all of the stuff is taken together, we have a better understanding of proteins and protein universe. Conclusions. We have intrinsically disordered proteins. They are present everywhere. They are easily detectable by computational tools. Intrinsic disorder is crucial for structure function continuum. These proteins are tightly controlled under normal conditions. If they're not controlled, it will be related to diseases. Some IDPs will be related to multiple different diseases. And all of the stuff will be coming back into this multifunctionality, multipathogeneity stuff related to proteoforms. We have not one protein, we have multiple proteins, multiple protein forms. And I'm done. Thank you.
Very much, Volodya.
So do you have questions?
Yes, are there any questions from the audience?
No questions. Okay.
No questions?
As usual, if you have some concept, it has two explanations. Explanation number one, it was so boring and not interesting that we were sleeping and we are not interested. And another possibility, it was cool and clean and we don't have any questions because of that.
It was cool and clean, but I have a question actually. Since this workshop was also about machine learning and AI in general and quantum computing, and we talked also about synthetic data creation in this workshop. So how would you suggest us to validate the synthetic data that we obtain for intrinsically disordered proteins? Which tools shall we use to validate our data?
Okay, so clearly it's not one.
Yes.
So my suggestion will be to take as many as you can. Because in this case, it will give you much better coverage. Also probably all computationals. It's an interesting question. What is more real? What is more reliable? Experimental work from wet lab or computational work from computer lab? If you're talking to experimentalist will tell you, unless you have experimental support, so all of your predictions are not real. In reality, if you look at how experiments are done, so they are not accurate by themselves. So you have also some issues over there. So while I'm telling you, so probably what you need to do, so you need to have one or two these synthetic molecules and try to see how they look like in wet lab, so to see how they behave. So, but obviously, even without this information, taking multiple disorder predictors so should help. So REDAO will be a good way to go. So you can try to use also some structure modeling tools. Like AlphaFold or modifications of AlphaFold. So for generating this stuff and see how the stuff look like.
Yes. Thank you very much, Volodya. There's another question. So.
Hello, Vladimir. Good morning. One more question. In your opinion, what is the best computational approach to compute time-dependent motion of the conformation of the IDPs? Any recommendation in how we should resolve this equation of motion computation?
Okay, so it's a good question. And motion will be reflected in coherent so distribution of positions. Yes.
Yes.
So I do like Afflector. Afflector takes AlphaFold and generates an ensemble. It's not the best way to look at it, but at least it's much better representation than AlphaFold. So because AlphaFold will give you just one structure, and what you need to have an ensemble of structures. But next question will be how accurate all of the stuff is, and so there is no any straight forward answer to it. So because even if you have data which are generated for your protein by experiment, like combination of SACs and NMR, it's not giving you an entire representation of entire ensemble. It gives you some experimental restraints, but it's not giving you a full picture. So now question will be how many different members of ensemble you need to include in your considerations in order to cover your stuff in the best way. And so there is no straight answer. It's incorrect question. It's an incorrect problem. So there is no straight answer to it.
Thank you.
So, but definitely, having one snapshot is a misleading. It's not an answer for sure. So which means that the structure which is generated by AlphaFold, it's not sufficient.
Yeah. And we came out actually with a way we discussed this in this workshop too, and we came out with a way to reduce the hallucinations in AlphaFold.
Yeah.
So
AlphaFold definitely has possibility to hallucinate for sure.
Yeah.
We'll take any protein which is involved in formation of long coiled structures. So AlphaFold will show you huge alpha helix in the middle, blue alpha helix with nothing else, which is not real.
Yeah.
Yeah.
Thank you.
Mm-hmm.
Are there any questions?
No questions. Okay. Thank you.
Thank you. Thank you very much, Vladimir.
Yeah, thank you.